Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a Bike. I'm going to give a quick shout out to a paper from the Binggui lab. So the other paper that I wanted to cover is Kheirbek lab. And this should feel parallel to paper covered in 15 or I think it's 14, where we looked at oligodendrous dendrogenesis and how that is coupled to remote memories of location in a water maze. So this paper is looking at fear conditioning, but is going to look at medial prefrontal cortex and the circuit changes that occur there. So maybe that makes this paper a little unique and a little special. In the first figure, they just go to show, the first figure shows that the recall of a fear memory, a remote fear memory, induces OPC proliferation. So these are oligodendrocyte precursor cells, and they begin to proliferate after after context fear conditioning. So after fear conditioning, oligodendrocytes divide. And the way that they show this is using that chemotherapy drug, EDU, that shows up in cells that have recently divided. And so they're just counting recently divided cells and looking at oligodendrocyte precursor cells to see how much cell division there is, and they show there's more cell division after context fear conditioning. And so they also show then in figure 2 that the OPCs actually mature into myelinating oligodendrocytes. So that's a separate step than dividing, right? You can have NG1 positive cells that are still oligodendrocyte precursor cells dividing without actually increasing any myelination. So figure 3 shows separately that those OPCs are also maturing, and that's important. So figure 4 shows that if they inhibit the formation of new myelin, then the expression of the remote fear memory is impaired. That's not very surprising. That's very similar to what Paul Franklin showed in the water maze experiments with blocking the conversion of oligodendrocyte precursor cells to oligodendrocytes. So a very similar result, but nice. I'm sure they were competing manuscripts at some point. So figure 6, I think, is going to look at immediate early gene expression and show that when they mess with myelin, then there's also impaired immediate early gene expression in the medial prefrontal cortex. So this is kind of like the molecular signature for activation and for learning. And so they demonstrate that during this period of reconsolidation, when the memory is solidifying, you need this myelination. Very similar to what they showed in the Paul Franklin paper. So figure 6, I think, actually doesn't. It shows calcium signals change in PFC. Figure 7 Figure 7 shows that if they over-myelinate, then they can see that fear memory gets generalized, and the immediate early gene expression increases. I think that's figure 8. So anyway, I'm not doing very well on this paper, but I do think I'm getting it mostly right. So that makes a nice complementary results from, I don't get it. I don't get how that's the way you drive. And now you're going to run me over. But it's a nice complement to the Paul Franklin paper, and it's a really cool sort of, parallel results are always nice in science. We don't get that very often. It's a different kind of memory. It's fear memory versus a water maze. And they do different ways of messing with blocking. But nonetheless, we're seeing it, right? I think the Franklin paper showed synchrony between PFC and hippocampus has changed. So, similar ideas. So the other paper that I wanted to cover is from the Yan Gu lab, and he's at a university in China, Zhechun, or I don't know how to say it, of course, and his group has shown something that Ben Barris and Beth Stephens have been arguing for quite a while now, and that is that microglia are responsible for synapse elimination, and that might be a very general mechanism throughout the brain. Well, this paper shows if they fear condition an animal and then look remotely, they see that the animals tend to freeze less after 35 days. That's pretty typical. That's just extinction. But what they look at is if they mess with microglia and deplete the microglia, that the fear memory actually persists better. And that is again registered by freezing. We don't have to like fear conditioning as a paradigm to see it as a very, very strong learning mechanism. And so it's worth studying it. But, it's obviously a little different than learning in the cortex, and it should be. But nonetheless, what they show is first in figure 1 that if they deplete the microglia, that they preserve more of this remote fear memory. That's really cool. And in figure 2, they show something I think is pretty extraordinary, and I'm not really sure if this has been shown previously. Could have been. But they show that microglia, via the complement system, and that's the complement cascade that causes apoptosis, etc. The complement system is what activates the microglia. And so in this figure, they use a molecule CD55 to interfere with the complement system. And if they do that, then they can show that, again, if the microglia are not able to mess with the synapses, then the fear memory is preserved. And so again, the remote fear memory is preserved if they mess around with the microglia. In this case, what they're doing is they're really eating synapses. And so in figure 2, what's cool is that they show synapsin molecules and PSD95 molecules inside of microglia. And that's pretty badass because they're eliminating the synapses. And I should say this paper is in the dentate gyrus. And so now what this paper is going to do is a kind of Tonigawa trick in that they're going to put Dredd, and that's an inhibitory designer drug. They're going to put these Dredds into engram cells. So the engram cells are the cells in the dentate gyrus that are activated by the context fear conditioning. And these are the cells that the Tonigawa lab has shown over and over again are coding the context of the fear. And if you mess with these cells, then you can mess with the manifestation of the fear response. So what he wants to do now is put a Dredd into the engram cells so that after training he can decrease their activity, essentially to demonstrate that without the activity caused by the initial training, then the complement cascade is not activated and the microglia don't prune the synapses, and therefore the fear memory is preserved. So that's a pretty cool way of doing it, and that's a pretty thorough way of doing it. I'm sold on this one. It's a really good one. Suffice it to say that they prove three things. First thing is they prove that fear memories last longer if they mess with microglia. If they mess with the complement cascade, they also have the same effect. So the idea is that the microglia, thank you, are activated by the complement cascade. And that without that activation, then they won't eliminate the synapses. So microglia are eliminating synapses. And the nice thing about this is that Ben Barris and Beth Stephens have argued this for a while. Beth Stephens has a new paper out which shows that her manipulation of the complement cascade with the C1q molecule doesn't really play a role in ocular dominance plasticity, which would be kind of contradictory to the idea that it's a general mechanism. It's been shown in the thalamus, and Beth Stephens has also shown it in Alzheimer's models, and that the synapse elimination in Alzheimer's models is played, the cascade plays a role in it, the complement cascade, and the complement cascade is activating microglia. So the question is really, what comes, how does this all work? Is it immune response, or is it really just sort of, synapses express different markers, and then those markers mark them for elimination? I mean, there's an entire sort of palette of molecular signals that, quite frankly, we don't know very much about because we don't pay much attention to microglia so far. And of course they're important, and of course they're going to be more important. The trouble is we don't know a lot about them. And it's very similar to the myelination thing. Myelination was just understood to be a maturation thing, something that was required in the adult brain, but the concept that it could be involved in memory consolidation or memory storage, or by messing with myelination you could eliminate long-term memory storage, that was also something I don't think a lot of people thought 10 years ago. So the next paper that I'm going to try and do is a paper that shows that even Hebbian plasticity, as we think about it classically, is not really operating in the hippocampus with CA1. So there's a lot of things changing in neuroscience, a lot of new ideas and a lot of new facets of the brain that are more important than we previously imagined. I mean, we're still at the stage where most of the brain cells are glial cells, and most of us still think they're just support cells, but that's clearly not the case, especially if myelination is integral to long-term memory storage, and if microglia and their activation are, god damn it. Okay, thanks for joining me on my ride. That were two papers involving fear memory and long-term fear memory. The first one is from the Kheirbek lab at the University of San Francisco, where they show that myelination is required. Not very dissimilar from Paul Franklin's paper that was also released recently. Instead of water maze learning, they're doing fear conditioning here. They show immediate early gene expression in the medial prefrontal cortex is also affected by myelination, downregulated when they eliminate myelination and upregulated when they overmyelinate. So that's a nice push-pull result. The other paper is from a lab in China, Yang Gu's lab, where they show that microglia are involved in eliminating context fear conditioning memory in the dentate gyrus. So specifically, they look at the engram cells and they look at synapses there. They show that microglia are actually phagocytosing synapses by showing that PST95 and synapsin are in microglia. They show that microglia is activated by the complement system by interfering with it, and they show that if they mess with the engram cells activity, then they can influence whether or not the synapses are eliminated, and if they mess with complement system or with microglia, then they can stop that process from happening. The main one being that if they use Dredds in the engram cells and then deactivate them or decrease their activity, then you get extreme forgetting, and that can be eliminated by the messing with microglia. So these two papers showing two different sides of fear memory, showing the necessity for myelination and also how memories might be eliminated by microglia. This is a general mechanism in the brain, at least as pushed by Ben Bars and by Beth Stephens, but Beth Stephens has a recent paper that I also just mentioned, which shows that maybe the complement system and microglia are not necessary for synapse elimination during the critical period in V1, which would kind of contradict the idea of it being a general mechanism, but there are problems with that paper and I didn't cover it, but let's just say that this is still up in the air, and I think microglia are much more important than we realize, especially for the purpose of eliminating synapses. If you liked it, like and subscribe, and I'll see you next time on JC on a Bike.